By the end of the COVID-19 pandemic, over 5.5 billion people, more than 2/3 of the human race, had received an injection of a technology that had never been authorized for long-term human use. Standard vaccine development typically requires 10 to 15 years of human safety testing to identify rare or delayed side effects. These mRNA platforms were deployed in a fraction of that time under emergency use authorization. This rollout was coordinated through global mandates. In many regions, the ability to work, travel, or participate in public life became legally tied to a person's participation in this medical program. To understand the implications of this shift, we have to look past the political debate and examine exactly what this mechanism does once it enters a human cell. The decision to prioritize speed over decades of clinical observation created a new operational baseline. This move established a model where public health crises can supersede the traditional requirements for long-term informed medical consent. The science behind this began in 1961. Researchers Jacques Monod and François Jacob hypothesized that a specific messenger molecule must carry instructions from the DNA in the nucleus to the protein-building machinery of the cell. In 1989, scientists at the Salk Institute achieved a breakthrough. They demonstrated that microscopic fatty envelopes, or nanoparticles, could deliver laboratory-made messenger RNA directly into living cells. The path to commercial use was stalled for decades because the human immune system naturally destroys foreign RNA on sight. Biochemist Cataline Carrico eventually discovered how to chemically modify the RNA to bypass these defenses, a discovery that later earned her a Nobel Prize. Carrico's modification provided the technical foundation for companies like Moderna and BioNTech to package and patent mRNA as a functional medical platform. While the core science was refined over 50 years in controlled lab environments, its application to billions of healthy humans represented a shift into an entirely new theater of clinical experimentation. The process begins with the lipid nanoparticle. This protective fatty coating allows the genetic instructions to slip through the outer membrane of your cells. Once inside, the mRNA moves through the cytoplasm. It does not enter the nucleus, which remains sealed, ensuring the instructions do not interact with your native DNA. The strand heads for the ribosome. This is the cell's protein-building machine, and the mRNA acts as a temporary set of instructions for it to follow. The ribosome reads the code and begins manufacturing exact copies of the SARS-CoV-2 spike protein, the specific shape the virus uses to infect tissue. After the proteins are built, the cell degrades the mRNA strand. The manufactured spike proteins are then displayed on the exterior surface of the cell. Your immune system detects these foreign shapes on your own cells. It flags them as threats and begins generating antibodies and memory B cells to recognize the virus in the future. This process effectively retools human cells to produce viral components. Deploying this technique on a global scale, without long-term tracking established, a population-wide biological baseline for an entirely new form of medicine. This chart tracks reports in the Vaccine Adverse Event Reporting System, or VAERS. Following the 2021 rollout, reports far exceeded the historical baseline of the previous 20 years combined. Within this data, researchers identified a documented increase in myocarditis, specifically concentrated among young males. Peer-reviewed concerns are also emerging regarding spikopathy. This theory suggests that modified spike proteins can persist in the body far longer than intended, potentially causing systemic inflammation or heart muscle impairment. Mainstream health agencies maintain that these proteins are harmless and quickly cleared. This widespread disagreement has resulted in a deep division within the scientific community regarding the long-term safety of the platform. Regardless of the final scientific consensus, the existence of these open questions confirms that the global rollout occurred while critical long-term data was still being collected. The mRNA platform is now expanding. Companies are developing universal flu shots, cancer therapies, and other genetic interventions using the same delivery mechanism. This technology is also entering the food supply. mRNA platforms are being developed for commercial livestock, including poultry, cattle, and pigs, to manage disease in industrial farming environments. This transition introduces a new variable, the impact of mRNA residues or synthesized proteins on the human food chain, a field where long-term safety data is currently non-existent. Beyond the biology, there is the precedent of normalization. The legal and social framework used during the pandemic suggests that gene-level interventions can be mandated as a standard requirement for participating in society. Protecting future choice requires demanding total transparency. This includes supporting legislation that ensures independent oversight and prevents emergency rules from bypassing standard protocols. The mRNA rollout shifted the boundaries of medical authority. It demonstrated that without active public oversight, the human body can be integrated into a new, mandatory infrastructure of corporate biotechnology.